Cuando ponemos Rivera hay que sacar este PowerPoint.
Hola Francisco, ¿me escuchás?
Sí, te escucho bien, Pablo. Hola,
¿Cómo va todo?
bien, acá estamos.
¿Todo bien?
Sí, sí.
Sí, me conecté hace 2 minutos, se escucha ruido de fondo.
Harto ruido de fondo.
Ajá, ya
Sí,
estamos en hora para empezar. Creo que vos sos el primero en el simposio, yo voy
sí.
segundo y después una postdoc de Janina es la última.
La última, sí. Problema que tengo es que al tuyo quizás me pueda quedar, pero después no me puedo quedar porque tengo otra cosa y
Ah,
urgente.
ok.
Ando
Ujú.
full. Pero
Francisco, Pablo, ¿nos escuchan?
ahora sí.
Sí, sí, sí.
¿Podemos conectarte, Francisco?
¿Ustedes nos escuchan a nosotros bien?
Sí, perfecto.
¿No escuchas?
Sí, perfecto, te escuchamos perfecto.
Eh, Avísame, ¿comparto la pantalla o...?
Compartí la pantalla, por favor.
Dale. ¿Ven ahí?
No, todavía no lo vemos.
Ok. ¿Ahora la ven?
No, todavía no. Avisamos.
Ok. A ver, voy a intentar de nuevo. Ahí lo ven, ¿no? Pablo, ¿tú la ves?
Sí, yo veo tu pantalla perfecta.
Perfecto.
Ahí estamos.
Y ahí se ve completa, ¿no?
Ok,
¿Se ve?
ponela entera. Ahí estamos, perfecto. Perfecto. Ok, thank you very much. After Dr. Macklin lecture, we'll have a symposium with 3 speakers. We are very lucky to have Dr. Francisco Rivera, originally from Chile, now in Finland. Francisco is professor of integrative neurobiology, translational regenerative neurobiology, and is a faculty of the biology and environmental science at the University of Helsinki in Finland. We will talk about something very interesting, that is circulating platelets with myelin repair and multiple sclerosis. They are free of course. So after that, we have Dr. Pablo Paez, somebody for our house. Pablo was one of the PhD students in Janina's lab, and after moved to do a postdoc in UCLA, from Campagnoni, and now is in Buffalo, New York. Finally, we have Vanessa Mattera, who was one of our wonderful PhD students, now doing a postdoc also in our lab, coming back from San Francisco to the States with the award, the pre-award from the International Federation of Multiple Sclerosis. And the talk of Francisco will be the very first. So thank you very much, Francisco, for accepting our invitation. So please go ahead.
Hi, thank you very much for this nice introduction. And I have to say that it's a pleasure to be here and to present what we have been working in the last years. And indeed, not only working here in Finland, but this work started when I was in Valdivia in Chile, and it was made through a collaboration with the laboratory of Robin Franklin in Cambridge, and then it ends up here in Helsinki. So it's a pleasure actually to show you a little bit about all the history and how that's started and how it ends. Thank you very much for the organizer. Thank you, Janina, also for this invitation. It's very, very nice to be here, because last time when was the first meeting about this Latin American GLIA club, I was not able to attend due to COVID, and now I have the chance at least to do it in a remote way. So thank you very much for that. Yeah, and what I will talk today is about the role that platelets have in the process of remyelination, and particularly in a context of multiple sclerosis. So of course, I will start here, as most of you probably already know. Myelin, it's insulating material that wraps around the axons from this multi-layer structure that which the main function is to allow the fast and efficient transmission of the impulses along the axons. It's mainly composed of 80% lipids, about 20% proteins, very specific proteins, and in the CNS, central nervous system, is produced by oligodendrocytes, while in the peripheral nervous system is produced mainly by Schwann cells. Now, myelin is a very fascinating structure of the central nervous system that has main functions. It's not just only this typical known function about allowing the fast and efficient transmission of the electrical impulse, which is already quite much, but in a way has been shown that also myelin allows metabolic coupling with axons that also promote them to survive better and adapt better to different conditions. And very important also is the fact that has been shown that myelin sheath is also important for motor skill learning. So bringing not only for a motor function, but also the cognitive output. So as you may know also, or many of you here, multiple sclerosis is an autoimmune demyelinating disease of the central nervous system that affects about 2.5 million patients worldwide. And it's a disease that indeed affects mainly young adults. It has the onset between 20 and 40 years old, most frequent, could be also older, and is the major cause indeed of neurological disabilities in young adults. And the symptoms varies, like double vision, weakness in legs, arms, cognitive disruption, which may include also loss of memory, and also promote fatigue and other kind of problems as well. Most of the treatments nowadays for multiple sclerosis focus on stopping the process of demyelination, so that most of them are immunomodulators. But however, they are not so the efficiency is not so high, and they have side effects. And very important for this talk today is that they lack reparative activity. And reparative activity is something that is quite nice in the central nervous system. At this, it also happens that myelin sheaths can be restored upon damage. So here I'm showing here in the gray color the neurons and the axons, and in purple here I'm showing an oligodendrocyte that is able to myelinate several axons, as you can see here. Upon demyelination in the central nervous system, we have two different scenarios. From one side, it's possible the myelin sheaths will never be restored, and this will lead to the axonal loss or axonal damage and death, which indeed leads to progressive decline in the neurological activity. However, nowadays, we know that myelin sheath can be also restored through a process known as remyelination. And here I'm showing a post-mortem forebrain section. As you can see here, this is a stain with the Luxol Fast Blue, and here you could see in the white color or in the unstained regions here, showing these lesions there, which are chronic demyelinated lesions. But also you can notice by these red arrows here, light blue, forming these kind of shadows. Well, these indeed are axons that suffer of demyelination, but already remyelination has been undergone. So indeed, what could happen also in the central nervous system is that myelin sheath can be restored, and new myelin sheaths normally has a smaller thickness compared to the myelin sheath that results from development. This indeed leads to functional recovery of the neurological activity, and this is the main importance of this process itself. Now, how does remyelination take place? Well, this is possible through the existence of oligodendrocyte progenitor cells that, of course, some or many of you already know about them. These cells are widely distributed through the central nervous system, white and gray matter. They represent a large population of glial cells, and they can be easily isolated and expanded in vitro. There are already known markers that allow us to do such expansions, such as A2B5, NG2, and PTGF receptor alpha, for example. OLIG1, OLIG2, which are transcriptional factors, very important also for oligodendrocyte development. Now, very briefly, sorry. Okay. So now, very briefly, what happens with the process of remyelination here? I will show you a very brief summary about the process how remyelination takes place. And you can see here the OPCs, and we also have astrocytes and microglia cells contributed to them. But this is just a very simple way to show the process. The process is much complex, but in a way to make it didactic and a little bit faster in this lecture, I'm trying just to show you a little bit a summary of how this occurs. Well, upon demyelination in the central nervous system, astrocyte and microglia cells get activated. They secrete neurotrophins and other soluble factors, growth factors that in a way promotes the recruitment of monocytes from the bloodstream towards the central nervous system. And in that way also, they're important on producing the myelin debris clearance. But also, they secrete soluble factors such as FGF or PTGF that in turn promote oligodendrocyte progenitor cell proliferation and activation. And then later, these cells are recruited towards the lesion site, where they will finally differentiate into remyelinating oligodendrocytes. So this is a short way to say how remyelination takes place. In our lab, we have three major aims. First, we aim to identify new unknown signals, factors, or cues that contribute to the process of remyelination, badly or wrong or goodly. So like contributing or impairing remyelination. This is important because the second aim is that we wanted to address is why remyelination fails in multiple sclerosis. And by the end of this, or the final goal in the lab, or one of the major final goals of our lab is to somehow figure out or to develop regenerative therapy for the treatment of multiple sclerosis. So these are the major three aims in our lab. And one of the projects in our lab that have been driven in the last, I would say, 4 or 5 years is the project about platelets. So platelets are small oval anucleated cells that are present in the bloodstream, and they are essential for forming the hemostatic plaque. This actually is the major function of platelets, but they also have others. They're mainly produced in the bone marrow by megakaryocytes, and very, very important is the fact that these platelets has these kind of organelles here that are stored compartments able to store a plethora of bioactive molecules, such as, for example, some of these storage compartments, the alpha granules. We also have the dense granules and the open canicular system in a way. These factors together not only contribute to the process of hemostasis, but also it has been shown important contribution to the process of inflammation, angiogenesis, and wound healing. Previously, like this was a study from 2015 or something like that, by then we studied a little bit about the interaction on platelets and neural stem progenitor cells present in the subventricular zone. And we found that upon central nervous system damage, platelets accumulate and promote progenitor cells survival under this context. So this actually tells us that platelets may have an important role during the process of repair or cellular turnover in the central nervous system. But indeed, what was very important data for us about what platelets may be doing in MS and in multiple sclerosis and EAE, for example, is the following studies. These studies are in postmortem, or actually not postmortem, but human biopsies of MS patients, and they were stained against CD41, which is a marker for platelets. And they found in these researchers that platelets accumulate in these demyelinated lesions or chronic lesions as well. However, in the normal opinion white matter of the same patients, there were no accumulation of such a platelets there. Other kind of studies also have shown that platelets may accumulate also in the experimental autoimmune encephalomyelitis animal model. So we could see here that 21 days upon induction of EAE, there is an accumulation of CD41 expressing platelets also in these plaques, but also in contact with microglia cells. However, this is not the only evidence that platelets may have an important role in the multiple sclerosis or in EAE. Indeed, other studies have shown an increased number of platelets or circulating platelets in EAE animal model. Also, other studies have shown that there is an important or a significant increase in the number of platelet-derived microparticles that are present in the MS patients. And together with that, studies have shown that these platelets display hyperactivity, which might explain also why you have an increase in the number of microparticles derived from platelets, and they display also an increased adhesiveness. So there are a number of platelet abnormalities in EAE or in MS that in a way suggest that we could study we should study platelets in the context of remyelination, and particularly in multiple sclerosis. So in collaboration with Robin Franklin's lab, when I was actually in Valdivia, as I previously mentioned, Amber Philp in our lab, she was a PhD student by then, she studied the potential role that platelets may have in the process of remyelination and whether and how they control oligodendrocyte progenitor cell function during this regenerative process. The first thing that she did then was to study the behavior of platelets in our animal model of focal demyelination. So what normally we do here, we injected lysolecithin in the white matter of adult mice in the spinal cord, and we evaluate the process of remyelination here. Normally what you could see 5 to 7 days post-lesion, that there is an increase or a peak in OPC, oligodendrocyte progenitor cell proliferation. At 14 days post-lesion, you normally found there a peak in OPC differentiation, and by 21 days post-lesion, normally you found that remyelination have ends. Now, what she did then study the presence of platelets at 1, 3, 7, and 14 days post-lesion, also 10 days post-lesion. So she used the marker CD41, the same one as we used in the previous studies, to detect the presence of platelets there. And what she found here, upon damage, is that at 1 day post-lesion, you already see some platelet aggregation here, and there is an increase on this aggregation at 3 days post-lesion, while thereafter 7 and 14 days post-lesion, there was significant drop down in the number of platelets. And she quantified that, and indeed she found that in that it was clearly the situation that 3 days upon damage, there was an accumulation of CD41 positive platelets there, and thereafter it drops down, even reaching the same levels as the normal appearing white matter. But of course, someone may think that if you are doing an injection in the central nervous system using a needle there that probably is injected inside of the central nervous system, you might induce mechanically wise the extravasation of blood-derived elements, such as, for example, the platelets themselves, you see. So this can be not due to a specific damage, but just the mechanical damage that is as a result of the injection itself. So to discard this possibility, Amber, what she did was that she injected PBS containing DAPI. So in a way, she was able to detect where the injection was, but instead of injecting this kind of demyelinating drug, she was just injecting PBS on there. And she found that where there were no specific aggregation of platelets in these lesion areas, or not lesion actually, like in these injected areas. So in a way, this is proof that there is a need of a particular damage, maybe not only myelin damage, maybe other kind of damage, that it indeed promotes the recruitment of platelets from the bloodstream towards the central nervous system. Then, as this is a demyelinated damage, the question is, what are the platelets then may be doing there? So she was checking for the interaction or the putative interaction between platelets and oligodendroglial cells, and she did that by 5 days post-lesion. And here in the green color, I'm showing CD41 expressing platelets. In the red, we can see collagen 4, which mainly stands for endothelial cells. And then you can see here the white nuclei as they are OLIG2 expressing cells, most likely oligodendrocyte progenitor cells, as this is a recently demyelinated region. And you can see here that platelets locate in very close proximity to oligodendrocyte progenitor cells here, which might indicate that these cells or these two cell types interact with each other. So to prove that, or to evaluate this possible interaction and the consequences of that, Amber used an animal model of platelet depletion. So what we normally do here is to inject this antibody against this glycoprotein 1 beta, which mainly leads to platelet depletion. And she did that 3 days after lesion. And then she injected this every second day until 14 days. She collected tissue by 5, 7, and 14 days post-lesion. And the first thing that she immediately noticed is that in the lesion site, she observed that platelet recruitment was significantly reduced in the lesion site in the platelet-depleted animal model compared to the untreated ones. So this is an appropriate model to evaluate such an effect. Then she checked for OPC differentiation at 7 days post-lesion, and she found that indeed there was a decrease on the number of differentiated oligodendrocytes. We use the stain against CC1 or APC in order to distinguish between mature oligodendrocytes and immature OPCs. she found a decrease not only on the total number of remyelinating or differentiated oligodendrocytes, but also in the percentage of these cells there in the depleted animal model. So after this, it's quite clear that platelet may influence directly or indirectly the OPC function in a way that is promoting, or in this case, platelet depletion may lead to a reduction in the percentage or in the number of mature oligodendrocytes. This may not necessarily need not may necessarily lead to a reduction in remyelination. So to check that, Amber did again exactly the same thing, or the same strategy, but then at 14 days post-lesion, she took the tissues and she embedded into resin and cut them into sections and then stained against toluidine blue to stain myelin. And then she evaluated by ranking analysis and quantify the percentage of remyelinating axons. And then she found, indeed, as you can see here, the untreated one at 14 days post-lesion, as you normally would find, there are a good number of remyelinated axons here, and you can see even in the percentage of remyelinated axons, but these remyelinated axons and the extension of remyelination get significantly reduced in the platelet-depleted animal model. So platelet depletion not only affects OPC differentiation, but also leads or hampers the remyelination extension and the percentage of remyelinated axons. Now, as someone may think, when you have such a situation in which you injected or you provoke platelet depletion, the vessels may become leakage, and in a way, it's quite possible that the vessels will become leakage, and that the blood elements will just drive into the parenchyma just because you have less platelets, right? So one of the major things, or one of the major blood-derived elements important for remyelination is indeed fibrinogen. And fibrinogen is important because it has been shown to inhibit OPC differentiation and induce atherogenesis. So what Amber did then was stain against fibrinogen, and we found no difference upon fibrinogen staining in the untreated compared to the platelet-depleted animal model, showing that indeed platelet depletion does not alter fibrinogen extravasation, and this way also do not alter atherogenesis. In other studies, we showed that atherogenesis was also not affected. Good, so we know that in our model, it might be the case that platelets may directly or indirectly promote OPC differentiation and then this way contribute to remyelination. But it can also be that remyelination, they contribute to remyelination through the modulation of macrophages, microglia, as we know already that platelets are important for inflammation, for the control and regulation of inflammatory response. So to evaluate that, Amber also stained against IBA1 microglia macrophages and pro-inflammatory markers such as CD1632 and arginase-1 as an anti-inflammatory marker. And she found here no difference at 10 days post-lesion in the untreated mice compared to the depleted mice. So there was no difference in the number of macrophages, microglia present in the lesion. There was no difference neither on the number of pro-inflammatory and anti-inflammatory macrophage, microglia present there. And you know that macrophage and macroglia are important for myelin debris clearance. So it could also be that platelets might be also involved in that. And in this way, by indirectly affecting or altering myelin debris clearance, it can promote OPC differentiation, because in a way, myelin debris is normally stopping OPC differentiation. So she checked this by staining against oil red O, and she found that indeed the clearance of myelin debris was not different in the untreated versus platelet depleted mice. So all this together suggests that most likely OPCs are directly affected by the presence of platelets and not indirectly through either fibrinogen stabilization or the regulation of inflammatory response. So in order to check and to study how the interactions may occur between circulating platelets and OPCs, What she did, she cultured OPCs in vitro and exposed them to complete wash platelets. And she did this in different fashions, in different models. As you know already from the in vivo studies, it's quite clear that platelets are there for a while, but then later on they are withdrawn from the lesion. So one approach is just to provoke or to expose OPCs transiently to platelets, to wash platelets. So in a transient manner, just a pulse. The idea was that the OPCs were exposed for 3 days in the to platelets, and then platelets were withdrawn, and we evaluate OPC differentiation at 6 days. And what we found there, indeed, is that upon different concentration of wash platelets induce the differentiation in vitro if the OPCs were just transiently or temporal exposed to platelets. However, what happened now if we just, instead of withdrawing platelets here, we left the platelets remain there all along the process of differentiation, such as what we see, for example, in the MS lesions, in which you still see the platelets present there in these demyelinated lesions. So if she did that, then she found that indeed this effect of wash platelets to promote OPC differentiation was neglected or suppressed. So if platelets remain there longer than what they should, this could actually suppress the pro-oligodendrogenic effect that they have on OPCs, which was quite surprising result for us, but very in line with the in vivo observations. Now the question is, what is producing this effect? Are the platelets alone producing that, or there is something else producing this kind of thing? So the hypothesis is that it might be platelet-contained molecules. As I previously mentioned, platelet has these storage compartments, which has a plethora of different bioactive molecules. So in a way, what we did, instead of just exposing the OPCs to the complete platelets, we expose OPCs to the platelet lysate. So we lysate the platelets and we just expose these OPCs to them. And we did it in both fashions, such as pulse manner or the sustained manner. And then what we observe here, upon exposure to these platelet-contained molecules, as expected, there was an increase in the number of differentiated oligodendrocytes. This was depicted just by the expression of myelin-basic protein here, and this was completely neglected or suppressed if the platelets or if the OPCs were exposed to platelets in a sustained manner, so the complete process. So this is actually as in the wash platelets, but now we know that something contained the platelets, that these cells that is promoting OPC differentiation only if OPCs are transiently exposed to them. Something that it was also interesting for us, and this was a question indeed from the reviewer, is what happens if now you do exactly the same thing as I was showing before, so you contain the platelets for the 6 days that we know already that the OPC's differentiation will be somehow suppressed there. But what happens now if we leave the OPCs for 3 more days and see whether then if the release of platelets will somehow rescue the OPC differentiation effect. And indeed, this is what happens. Platelet withdrawal rescues the capability to promote OPC differentiation. So even though we expose OPCs to sustained manner for 6 days, if platelets are withdrawn of the media, then these OPCs will rescue or will somehow rescue the capacity to differentiate better, which is interesting. Very interesting. But then the question is, well, we already show in vivo before that platelets may not affect microglia macrophage differentiation and macrophage-macroglia polarization, right? So then we did exactly the same thing now just to confirm these findings in vivo, and we found, or we exposed BV2 microglia cells to platelet lysate for different time points, 24 hours, sorry. And then we found that by ELISA and by PCR that indeed the exposure to platelets BV2 cells do not really affect their polarization stage. That means that this is very in line with our in vivo result, findings. Altogether suggests that platelets modulate directly OPC function in a bimodal manner, because if they are exposed shortly to platelets, these OPCs will be increased their differentiation stage, but if they are exposed permanently or in a sustained manner to platelets, OPC differentiation will be affected. So let's see now. We just focus now on OPCs because it's quite clear that OPCs are these ones, the cells affected. But instead of using all in vitro approaches, we came back to an in vivo approach, and we use an animal model which allow us to evaluate the consequences of a thrombocytosis, an increase in the number of circulating platelets, but a sustained increase in the number of circulating platelets. So we use an animal model that has a mutation in the calretinin gene under the control of this hematopoietic marker, which is called the VAF. And this animal model, under heterozygous condition, it promotes somehow an increase in the number of circulating platelets without affecting other hematopoietic cells, lineage cells. And the first thing here is that she did a lesion in these animal models. And the first thing that she checked here was the number of circulating platelets upon the lesion. So this is circulating. And she observed that indeed there was an increase, always sustained increase in the number of circulating platelets that were present in this animal model. Now, if she went back and checked what happened with the lesion itself and the recruitment of platelets to the lesion itself in this thrombocytosis animal model, then she found that indeed in this, which is we call them CALR, she found that was an accumulation of platelets 5 days post-lesion in the lesion, and this accumulation was sustained even after 10 days post-lesion here. So it was a reduction between 5 and 10, but the accumulation was always higher than compared to the wild type. So this is a very nice animal model to prove that, or to test at least, whether in vivo a sustained increase in the number of platelets or a sustained recruitment of platelets to the lesion may also hampers the process of remyelination and OPC differentiation. Indeed, this is what she showed later, that in this thrombocytosis animal model, the total number of oligodendrocytes or OLI2 positive cells was not affected. However, the number of mature oligodendrocytes was significantly reduced in this animal model, as well as the percentage of these differentiated cells. And very nicely, she evaluated the correlation between the number of circulating platelets and the generation of new remyelinating or mature oligodendrocytes, and she observed a good or a positive correlation among these two things. So, by end this presentation, I can say that platelets early react to demyelination and aggregate, locating in close proximity to oligodendroglial cells. Platelet depletion leads to a reduction in OPC differentiation and hampers remyelination. Also, platelets promote OPC differentiation. However, if the exposure is sustained, platelets suppress these effects. And these effects are mainly mediated by platelet-contained molecules. A sustained thrombocytosis in vivo animal model also show alterations, a decline in OPC differentiation during remyelination. And altogether, these findings indicate of a beneficial role of platelets in remyelination. However, an abnormal sustained platelet activity, such as what happens in multiple sclerosis, might be detrimental for myelin repair. And this is why here I'm depicting the Dr. Jekyll and Mr. Hyde role of platelets, this bimodal way that platelets may play a role. They are good if they are in the correct moment and for the correct time there, but it's not good if they remain there longer than what they should. So in a way, this could hamper the process of remyelination, and this may be a reason that by which is contributing to remyelination failure in multiple sclerosis. I would like to finalize my talk just to say thank you to all our collaborators. This is part of my members lab here. These are former labs in Universidad Austral de Chile. Our collaborators here in Helsinki, and also what we find abroad. And of course, I would like to thank all the support that we have been receiving for the last 5 years that make this study possible. So thank you very much.
very much, Francisco. We have short time, so we have time for 2 short questions, 2 short answers. Dr. Reed? Francisco.
Very nice work, Francisco. Have you analyzed transcriptomic databases for platelets, and what do they show?
Sorry, could you repeat? If I have is what?
Available transcriptomic databases.
I couldn't hear very well. Sorry for that. Is the question about the contents of platelets? Hi? It's a question about the contents of platelets. If I evaluate what is inside of the platelet, that's the question? Jorge, can you listen me? Jorge, can you listen to me?
Do you have transcriptomes of your blood?
No, we are now working on that in a way. But first of all, we are trying to discard certain factors that might be obviously present there, such as, for example, PDGF. That's what's one thing. And the transcriptome, we were now working on that, but not just evaluating the transcriptome of healthy platelets, but we are now evaluating also platelets on multiple sclerosis patients, and then comparing those.
Thank you.
But one of the factors is PDGF receptor alpha, which PDGF AA, which is quite obvious.
Francisco, can you hear me?
Yes, now I can hear you.
Two quick questions. First of all, I didn't get how the platelets get to the MS lesion, the extravasates. And the second question is, if you know what is the activation states of these platelets, are they activated in a way, or they are just regular platelets without any secretion?
It's very good questions, actually. The first question is about how they are recruited to the lesion, right? So the thing is that it's not really known how the platelets might be recruited, because it's supposed to be not just a travasation, like you have the flux of the blood and that's it, that's going mechanically there, right? But what it's supposed to be here, and this is something we are also now addressing in the lab, is if there is any kind of chemoattraction or something like explain the recruitment of platelets, because this happens between 1 and 3 days, right? It's not happening immediately. And there is an active effect or an active signal coming out from there. And we are trying to check what is the real mechanism occurring there. It's not just injection, as I said, but there is a potential recruitment of platelets that might be done by a certain mechanism. And it's not much known about that overall in general. That's one thing. And the second thing is that, well, you have always the activation of platelets, P-selectin, and then you have the interaction in the endothelium, and then you have the travasation. But how this is then later on going through the central nervous system, this is not known. Now, concerning the question about the activation, we did CD62P stainings, and we found that the cells were also positive for that.
Thank you very much, Francisco. Wonderful talk. happy that you accept our invitation. So hopefully next GLIA class meeting we will be here. Thank you. Thanks a lot. Okay, our next speaker is Dr. Pablo Paez. Pablo is professor of pharmacology and toxicology at the University of Buffalo in New York. He, as I mentioned before, he is far away. He's part of our house. So Pablo will talk about chemiogenic manipulation of oligodendrocytes development and function. Thank you very much, Pablo, for accepting our invitation.
Oh, thank you very much, Jorge, for your invitation, and Janina, also Patricia, for organizing the event. So can you hear me well? Okay.
Yes.
So basically today I'm going to talk about calcium signaling in schwann cells, in oligodendral cells. So we know that calcium signaling are essential for OPC differentiation, for OPCs to respond to the electrical activity of axons. So we know that there are calcium changes in the soma of the OPCs during development and also in the tip of the processes. In the adult brain, calcium signaling is also very important for myelin maintenance and remodeling, cell survival, myelination, and axonal metabolic support. And it's also very important in pathological conditions. For example, it's a key step in excitotoxicity cell death. So we know that in vivo and in vitro, there is calcium signaling happening in OPCs, and oligos are essential for the development and the maturation of these cells. But the calcium channels and the receptors involved in these effects are not very well understood. So, for example, we know that in the tip of the processes during OPC development, there is high concentration and activity of voltage-gated calcium channels, for example. Calcium channels that respond to glutamate, like AMPA and NMDA, and calcium channels that respond to ATP, also GPCRs. We found a couple of years ago that, for example, voltage-gated calcium channels, the activity of voltage-gated calcium channels are very important for the maturation of OPCs, for the migration of these cells during development, and also for the proliferation of immature cells. So the hypothesis in our lab is basically that using all these calcium channels and receptors, OPCs sense the electrical activity of neurons, particularly the release of neurotransmitters like glutamate, ATP, and GABA, and they transduce this electrical activity in calcium signaling, which are very important for maturation of the cells. So to understand how OPCs respond to the electrical activity of neurons, and particularly to different group of neurotransmitters, we are doing some in vitro experiment using the pseudopod subcellular fraction system. So basically, we are growing OPCs on top of the membrane that have small holes, small pores, from which OPCs they can extend processes. So we are treating these cells with glutamate for 3 days, and we can isolate the soma of the cells and the processes of the cells. So this is how the chamber look like: top view, bottom view. In the top we have the cell soma, nuclear, DAPI staining, and then in the bottom pure process. So after growing the cells in the system for 2 days in vitro, 4 days in vitro, we can isolate the soma of the cells and the processes independently and check the expression of proteins and messenger and see how the processes of these cells respond to different neurotransmitters. So, for example, these are one of the first experiments that we did in the system. These are real-time PCR, qPCRs. And we found, for example, that OPCs that were treated with glutamate responds with upregulation of different proteins that are known to be involved in the formation of synaptic connections, like, for example, ACAP 150, Homer, PSD 95, and SHANK. Some of these proteins they are upregulated, and also they are apparently moving from the body of the cells to the processes. Like you can see here, for example, for ACAP 150, Homer, and Paez identified. There is a upregulation in the presence of glutamate compared to cells in basal conditions. And actually, the expression of these proteins or the messenger for these synaptic proteins is highly upregulated in the process of OPCs that will grow in the presence of glutamate. So we isolate total RNA from the processes of these OPCs in this chamber in vitro, and we conduct whole RNA sequencing. And we found that glutamate, for example, increased the presence of messengers of genes related with synaptic pruning, plasma membrane function, central nervous system myelin maintenance, positive regulation on cell-cell additions mediated by integrins, and also oligo development. So if we put all these data together, glutamate is stimulating not only the extension of processes in OPCs, it's also upregulating the expression of proteins related with synaptic connections and cell-cell addition, in addition to genes related with oligo differentiation. We're also doing proteomics in this system. We are analyzing the protein content of OPCs processes that grow in the presence of glutamate. We found more than, if we put all this together, is more than 5,000 proteins specifically expressed in the processes of OPCs that respond to glutamate. We found approximately 482 proteins that were downregulated with the presence of glutamate in these cells, and 400 proteins that were upregulated. So if we combine the expression of these proteins in biological processes of cellular components, for example, we found that proteins related with cell-cell additions, messenger RNA processing, metabolic processing, proteins transport, and focal addition, extracellular exosome, cytoskeletal are upregulated by glutamate in the processes of OPCs. Of course, we found upregulation of several cytoskeletal proteins like tubulin, actin, RAC, for example, so suggesting that glutamate, right, this neurotransmitter, is promoting the extension of processes of OPCs, and also postsynaptic components, like I showed in the previous slides, by qPCRs in the process of these cells, like ACAP5, Homer, and also CAM-kinase2. We are also doing analysis of how these proteins are interacting between each other in the process of OPCs, and we found, for example, using the inhibitory pathway that, for example, Homer is central in this protein complex in the process of OPCs, and is interacting with CAM-kinase2, and also with several others postsynaptic proteins. In vivo, we found that, so to confirm these results that we found in this chamber in vivo, we did RNA sequencing from OPCs isolated from the cortex of P10 and P15 animals. So we did RNA sequencing, and we focus our analysis in calcium channels, receptor, calcium signaling, and postsynaptic proteins. And we found, for example, that in cortical OPCs between P10 and P15, there is a significant upregulation of, for example, voltage-gated calcium channels, AMPA receptors, NMDA receptors for glutamate. Also, GPCRs that respond to glutamate, like mGlu5, mGlu23, and also, again, several postsynaptic proteins like PSD95, SHANK, Homer, and proteins related with calcium signaling and calcium release from the ER. So, putting all these results together, the RNA sequencing and the proteomics that we are doing in the process of OPCs, in the Boyden chamber in response to glutamate, and also this in vivo RNA sequencing. Our working model is like, look like this, right? Basically, in the tip of the process of OPCs, we have a high concentration of voltage-gated calcium channels, AMPA receptor, and NMDA receptor, GPCR for glutamate, and all these receptors and channels are associated with postsynaptic proteins similar to what is going on in neurons, being central PSD95, Homer, CAM-kinase2, And also all this complex is associated with the cytoskeletal network. So we believe that these calcium channels are receptors are essential for OPCs to sense electrical activity of neurons to respond to the release of neurotransmitters like glutamate. We are doing now similar experiment with ATP and GABA to see if there is difference in the expression of these calcium channels and receptors, and also the postsynaptic proteins in response to these particular neurotransmitters. But we believe that this is one of the first complex that is established in OPCs when the cells are migrating and proliferating, and is first establishing contact with axons. So basically, we believe that OPCs are transducing the electrical activity of neurons by calcium signaling using these channels and this complex. So in the second part of my presentation, I'm going to talk about REITs. This is a new technology. These are designer receptor exclusive activated by designer drugs. These are receptors that can be activated specifically by drugs like CNO, clonazepine, right? CNO is an inert metabolite of clonazepine. So basically, these are very nice tools to modulate the calcium signaling in cells and also the electrical activity of cells. For example, the M3 is an excitatory GPCR that is being used for activate neurons to increase the action potentials and the electrical activity of neurons, and the inhibitory M4 is being used to reduce the electrical, the excitability of neurons and electrical activity of neurons. So we are using this technology, M3 and M4, excitatory GPCRs, inhibitory GPCRs, to modulate calcium signal in OPCs during development and see how these changes affect the development of these cells. For example, M3 is known that to increase intracellular calcium concentration in neurons, increase the activity of MAP kinesis and the activity of voltage-gated calcium channels. And then M4, they have the opposite effect. They reduce the activity of voltage-gated calcium channel in the plasma membrane of neurons. They increase the activity of inward rectifying potassium channels, which are essential to maintain the action potential, the electrical properties of the axons, and also increase MAP kinase activity. So we are doing calcium imaging in vitro in primary cultures of cortical OPCs. So this is, for example, how OPCs respond to glutamate. As I show you in the first part of my presentation, OPCs express high levels of NMDA and AMPA, and also GPCRs for glutamate. As you see, the first flash is the activation of probably calcium permeably NMDA receptors in OPCs, and then the flickering of the cells is reflecting the activation of metabolic GPCRs for glutamate. OPCs in vitro are also highly responsive to acetylcholine, as you can see in this video. So basically now we are expressing M3 in the cells, and we are checking how the activation of this excitatory GPCR is affecting calcium signaling in OPCs in vitro. So these are our preliminary results. For example, number one, we found that if we treat OPCs with CNO, this is the drug that activates the M3 receptors in OPC. These are cortical OPCs coming from transgenic animals that express M3 specifically in glial cells. If we treat these cells with CNO in vitro, there is a significant release of calcium from the ER. Number one, this suggests that M3 is being expressed in OPCs. And number two, that the receptor is functional. And then we, after treating the cells with CNO for 30 minutes, after activation of M3 for 30 minutes, we check how this affects the activation and the activity of different groups of calcium channels. So the first experiment that we did was stimulation with high potassium. This depolarized the plasma membrane of OPCs and induced calcium influx via voltage-gated calcium channels. You can see cells that were pretreated with CNO show a significant increase in calcium influx after stimulation. We found similar results after treating the cells with glutamate. We found a reduction in the activity of ATP calcium channels, and also see pointer. Sorry. Here we go. And we found a reduction after ATP stimulation and no changes after acetylcholine stimulation. So this suggests that all these together, that the activation of these excitatory GPCRs in OPCs, the M3 receptor is increasing, like in neurons, the activity of voltage-gated calcium channels, the activity of glutamate channels is reducing the response of the cell to ATP, but it's now changing the response of OPCs to acetylcholine. So basically, the activation of the receptor is changing the modulation, the dynamic of calcium signaling in OPCs in vitro. Then, using the same system, these are primary cultured cortical OPCs isolated from a transgenic animal that express the M3 receptor specifically in OPCs. We follow the differentiation of the cells in the presence of vehicles or in the presence of CNO. Again, CNO is the drug that activates the M3 receptors in OPCs. And we found that these, after 3 days of CNO stimulation or treatment, the percentage of OLIG2 positive cells expressed in SOC2, which is a marker of immature oligos, was significantly increased. Also, the number of cells expressed in NG2 was upregulated in the presence of CNO. We found a clear increase in OPC proliferation, so suggesting that activation of N3 is promoting OPC division and mitotic activity in these cells. No changes in cell death, so the activation of this M3 for 3 days in vitro is not inducing cell death. And then we found a reduction in the number of CC1 and PLP positive cells, suggesting that it's a significant reduction in the maturation of these cells. So again, all this together is indicating that activation of N3 in primary culture of OPCs is promoting the proliferation of these cells, is keeping these cells in an immature state, and is reducing the ratio of maturation. Moving in vivo, we found, again, using the same transgenic animals in which we are expressing the M3 receptor. This is the excitatory GPCR, specifically in SOX10 or NG2 positive cells. We confirmed that more than 99% of OLIG2 positive cells are expressing the receptor in vivo. We use this protocol. This is an inducible conditional knockout. So basically, we are treating these animals with tamoxifen between P2 and P6 to induce the expression of the N3 receptors in immature OPCs during the first postnatal week. And then we are injecting daily all these animals from P10 to P20 with CNO to activate this receptor in vivo. So one of the first experiments that we did were calcium imaging in tissue slices from these animals. Again, if we treat these cells, the M3 positive cells, with CNO, there was a significant increase in intracellular calcium concentration. The shape of the response suggests that this is calcium release from the ER. And similar to what we found in primary cultures of cortical OPCs coming from these same transgenic animals, there was an upregulation of the activity of voltage-gated calcium channels, glutamate channels, a reduction in the activity of ATP, and no changes in the response to acetylcholine of these cells. So again, in vivo, as well as in vitro, the activation of N3 is changing, the activity of different group of calcium channels is changing, the calcium signaling in these cells and how these cells respond to different neurotransmitters. So the question is how these affect the development of OPCs early in the brain. So using the same protocol, again, with tamoxifen, we are inducing the expression of the M3 receptors in OPC between P2 and P6. Then we treat these animals between P10 and P20. This is the peak of myelination in rodents with CNO to activate the M3, specifically in OPCs. Doing immunohistochemistry for myelin proteins. This is PLP. And this is MVP. We found significant reduction in the expression of on the synthesis of myelins in the cortex, in the striatum, in the lateral corpus callosum, and also in the central corpus callosum, suggesting that the myelination, the postnatal myelination, of animals in which the M3 receptor is being activated in OPC significantly delay. We confirmed this data with black hole staining for myelin. As you can see, there was a significant reduction of myelin proteins of myelin in the central portion of the corpus callosum, in the cortex, and also in the striatum. We confirmed that with electron microscopy. In the body of the corpus callosum, we found a significant reduction in the percentage of myelinated axons and also an increase in G ratio. And this was also clear in Western blot experiment that were completed with proteins isolated from the cortex and the cerebellum of vehicles and CNO-treated animals. You can see the significant reduction in the expression of MVP, CMP, and PLP. So the activation of the M3, these excitatory GPCRs in OPCs, is delaying the myelination of the postnatal brain. We check the population of OPCs in these animals. We did immunohistochemistry for the M3 in combination with CC1 or LIC2. We count number of cells in different areas of the brain. This is the result from the cortex. This is the result of the lateral corpus callosum. In agreement with a reduction in the expression of myelin proteins, we found that activation of M3 in OPCs early in development is reducing significantly the number of CC1 positive cells, which are post-mitotic premiyelinated and myelinated oligos in both the structure, in the cortex, as well as in the lateral corpus callosum. We found an increase in the number of M3 positive cells. This was interesting. If we calculate the percentage of M3 cells expressing CC1, so just to check the ratio of maturation of cells expressing M3, we found a significant reduction. So suggesting that the maturation of cells expressing the GPCR, the excitatory GPCR M3 in the presence of CNO is reduced, right? And in agreement with what we found in vitro, in primary cultures of cortical OPCs, we found a significant increase in the number of proliferating cells. So we checked that specifically in M3 positive cell population. As you can see across development, P15, P20, P30, there was a clear increase in the number of M3 positive cells expressing Ki-67, which is a marker of mitotic cells. And in agreement with that, the percentage of M3 expressing SOX2, again, a marker of immature oligos, was upregulated at P20 and P30 in our CNO-treated animals compared to animals injected with vehicle. We did also RNA sequencing from cortical OPCs from these transgenic animals, again, expressing the M3 specifically in OPCs in NG2 positive cells. And in agreement with the immunohistochemistry data and also with the immunos in primary cultures of OPCs, we found that there is a significant upregulation of genes and processes related with cell proliferation. That, for example, structural constituted of chromatin, cyclin-dependent protein serine kinase, regulation activity of chromosome segregation, cyclin-dependent protein kinase complexes, DNA packaging complexes, all genes related with these processes, basically DNA duplication, were upregulated in cells treated with expressing M3 and treated with CNO. We found also a downregulation of genes related with cell recognition, receptor ligand activity, and signal receptor regulatory activity. So basically, if we put all these together, right, the activation of this excitatory DRID, this excitatory GPCR, specifically in OPCs in the cortex during development, is reducing OPC differentiation, is delaying the myelination of the postnatal brain, and is promoting the proliferation of these cells, right? Mitotic cell cycle checkpoint signaling upregulated, regulation of chromosome segregation upregulated. So in vitro, in vivo, and also this ex vivo experiment with isolated OPCs is indicating that the calcium changes that this excitatory drip is producing in OPCs is affecting significantly the maturation of these cells. Then in the next set of experiment, we check the effect of activating M3 specifically in OPCs of the adult brain. So for this experiment, we use the NG2 and 3. So we are expressing the M3, the human DRID, M3 receptor, specifically in OPCs in the adult brain. We inject tamoxifen between P50, P55 to specifically express this receptor in adult OPCs, and then we treat these animals with CNO to activate the receptor between P60 and P70. So basically in this protocol, we found no changes in myelin, so activation of M3 in OPCs in the adult brain is not affecting the myelination of the adult brain, but we found, and also there were no changes in the density of mature post-mitotic oligos, CC1 positive cells, but we found, similar to early during development, an increase in the number of M3 positive cells, and the percentage of M3 succinate positive cells, and also an increase in cell proliferation. So suggesting that similar to what happened in OPCs during development, the activation of this treat in OPCs in the adult brain is promoting the proliferation of OPCs. Then we did the same experiment, but using the SOX10 treat. So this was to activate, to express the M3 receptors in mature myelinated oligos. In this case, we found a significant reduction in the expression of myelin proteins, so suggesting some demyelination in these adult animals. Also, this was associated with a reduction in the number of CC1 positive cells, small changes in the number of cells expressing M3, but significant reduction in the number of M3 positive cells expressing CC1, and also in the number of M3 cells expressing caspase-3, an activated form of caspase-3, suggesting that some of these mature oligos are dying by apoptosis. If we put all this together, we confirmed that with electron microscopy in the central portion of the corpus callosum. So at P90, we found a significant increase in the G ratio, reduction in the number of myelinated axons, and a significant also increase in the axons with abnormal myelin and signs of axonal degeneration. Western blot also are confirming this hypomyelination. As you can see here, the number of axons with sign of demyelination and also degenerations are increasing the CNO treatment. So basically, yeah, the activation of, we found, right, this putting all together, the activation of this excitatory GPCR, this excitatory DRIDs in OPCs during postnatal development. This is in vitro and also in vivo, induce ER calcium release, the release of calcium from endoplasmic reticulum. This increase, again, this is we found this in vitro and in vivo, increased activity of glutamate and voltage-gated calcium channels, and this promote OPC proliferation and significantly reduce OPC maturation. So this is delaying the myelination of the postnatal brain and also the maturation of cortical OPCs in vitro. The activation of M3, specifically in adult OPCs, we found that this increased OPC proliferation similar to OPCs early in development and have no effect in myelin. But the activation of M3 in myelinated mature postmitotic oligo apparently is toxic, induced demyelination, reduce the number of postmitotic oligos, and this is affecting myelin and is inducing demyelination of the central nervous system. So then in the last couple of slides, I will show you some data that we have using the inhibitory DRID, the M4 in OPCs. Again, these are primary culture of cortical OPCs isolated from a transgenic animal expressing the M4 receptor, the inhibitory DRID is inhibitory GPCR in OPCs. In this set of experiments, we found that activation of this receptor by CNO is significantly reducing, opposite to what we found with the M3, is reducing the activation of glutamate channels and receptor, is reducing the response of the cell to ATP after 30 minutes of the stimulation or after 3 days of stimulation with CNO, and is also reducing the activation of voltage-gated calcium channels. So similar to similar to the effect of this inhibitory treat in neurons, and in opposition to what we found with activation of the excitatory treat in OPCs. We also follow the maturation of the cells in vitro after 3 days of CNO stimulation. Similar to what we found with M3, we found that the percentage of OLIG2 positive cells expressing NG2 was increased. The percentage of the cells expressing SOC2 also was significantly increased. There was some increase also in proliferation and a reduction in the number of post-mitotic CC1 positive cells, suggesting that the activation of M4 in primary cultures of OPCs is also reducing the maturation of these cells in vitro. In vivo, there was a mixed effect. At P20, we found hypomyelination. We checked the expression of MVP and PLP in the cortex, in the corpus callosum. As you can see here, there was almost 50% reduction, right, using this protocol again, in activation of the M4 receptor between P10 and P20, significant reduction in the expression of myelin proteins. But at P30, right, 10 days after the last CNO injection, we found upregulation of myelin protein expression in different areas of the brain. This was also cleared by black hole staining, more myelin in the cortex, in the corpus callosum, and also in the striatum, and the same effect of B60, right? So this suggests that initially the activation of M4, this inhibitory treat in OPCs, is delaying transiently the myelination of the postnatal brain, but then these animals catch up, and then eventually they have more myelin that cells treated with vehicle. We check the population of OPCs in this system using the same protocol. Again, this is an inducible conditional knockout, so conditional knock-in. So we are inducing the expression of M4 in NG2 positive cells, and sorry, in SOX10 positive cells between P2 and P6, and then we are activating the receptor M4 between P10 and P20. There was a transient reduction in number of CC1 positive cells at P20, but P30, P60, number of post-mitotic mature oligos was completely normal. The percentage of OLIG2 expressing CC1 was also reduced initially, but then there was normal in the cells. But similar to what happened with the activation of M3, the excitatory drip in OPCs, the number of mitotic proliferating OPCs was increased across development. The same happened with the number of cells expressing M4, suggesting that this population of OPCs expressing the inhibitory drip is expanding in the brain. There was also an increase at P30, P60 in the percentage of M4 expressing CC1. This is in agreement with more myelin. We found more myelin in these animals, particularly P30, P60. And the number of M4 expressing Q67. So the proliferation of cells, specifically expressing the M4, was also increase. So this was very interesting. Similar to what we did with M3 in the adult brain, we express this inhibitory drip in mature post-mitotic oligos using the SOX10 line. We inject tamoxifen between P60 and P65 to specifically express the receptor in mature myelinated oligos. We treat these animals with CNO between P70 and P80, and then we check the brain at P90. And opposite to what we found with M3, M3 was inducing the myelination, axonal degeneration, and it was reducing the number of mature oligos. The activation of this inhibitory drip in mature myelinated oligo is promoting the synthesis of myelin, is increasing the expression of MVP and PLP in the adult brain at P90 without affecting the population of oligos. The number and the density of OLIG2 in the cortex or in the corpus callosum was normal, CC1 positive cells the same. We found no changes in cell proliferation, so everything was completely fine. but the expression of myelin proteins was significantly higher in these animals. And these are Western blot showing, confirming these results, a P30 and P90 activation again of this inhibitory drip M4 in OPCs in the adult brain is promoting the synthesis and the expression of myelin proteins. Now we are doing electron microscopy experiment in these animals to confirm, to check the ultrastructure of myelin and to check the percentage of mediated axons and the myelin morphology. So conclusion for M4 activation in OPCs early during development, opposite to what we found with the excitatory drip, the activation of M4 in OPCs reduce calcium influx through ATP, glutamate, and voltage-gated calcium channels, promote OPC proliferation and delays OPC maturation early in development, and decrease myelin synthesis, initially at P20, but then promotes myelin in young adult brains. The activation of 4 in myelinated oligos increase myelination of the adult brain and the synthesis of myelin proteins, opposite to what we found with the M3 receptors. So basically, in terms of calcium signaling, excitatory drip is promoting calcium signaling in OPCs, the activity of glutamate channels, the activity of voltage-gated calcium channels. Inhibitory drip is reducing ATP, glutamate, and voltage-gated calcium channels activity. So we believe that these calcium changes are significantly affected the maturation of OPCs. In both cases, we found that early in mature cells, this promote proliferation and delay the maturation of the cells. But the M3 basically is reducing OPC maturation and oligo development through and all the time points that we check. But the M4 is having the opposite effect, particularly in the adult brain, is promoting the synthesis of myelin proteins. So finally, in the last 3 slices, we show some preliminary data that we have in Schwann cells. Apparently, this is also a nice system to modulate calcium signaling in Schwann cells. These are primary cultures of DRGs expressing the M3 receptor. Again, like in OPCs, the activation of this receptor, this excitatory GPCR is inducing calcium release from the endoplasmic reticulum, and is significantly changing the activity of different group of calcium channels in Schwann cells. We found that this M3 activation in Schwann cells is reducing the activity of ATP, as reducing the activity of acetylcholine with and without calcium. So this suggests that there are ionotropic and metabotropic channels being affected. Similar to what we found in the central nervous system, in the peripheral nervous system, this is the sciatic nerve. The activation of M3 in Schwann cells is significantly delaying the maturation of Schwann cells and the myelination of peripheral nerves. This is electron microscopy showing a significant reduction in the number of myelinated axons at P10, P15. Here we are injecting CNO between P6 and P15. Changes in the shear ratio were very clear or across development, P10 and P15. Western blot confirming that a significant reduction in the expression of myelin proteins in the peripheral nervous system. So early during development, activation of this excitatory drip in Schwann cells was significantly delaying the myelination of peripheral nerve. In the adult peripheral nervous system, again, for this, we use the same protocol that we use for the central nervous system for oligos and OPCs. We inject tamoxifen between P50, P55 to induce the expression of the M3 receptor, specifically in Schwann cells, in adult myelinating Schwann cells, and then we stimulate the cells between P60 and P70 with CNO to activate the receptor. In agreement with what we found in myelinated oligos, the activation of this excitatory drip, they induced demyelination of peripheral nerves, and a significant increase in the shear ratio, reduction in the percentage of myelinated axons, significant also increase in the number of axons with a normal myelin and signs of neural degeneration. Also, there was a significant increase in the number of amyelinated axons associated in bundles in animals in which the M3 receptor is being activated in Schwann cells, Western blot confirming this demyelination, and also this is significantly affecting motor coordination in animals. These are rot experiment showing that after 10 days of CNO treatment, after 10 days of being activating M3 receptor in Schwann cells, this demyelination of peripheral nerves is significantly affecting the motor coordination of these animals. So in the peripheral nervous system, apparently this is also a nice tool to manipulate the maturation and the development of Schwann cells. We found in vitro in primary cultures of DRGs that the activation of this excitatory drip in Schwann cells is inducing the calcium release from ATP, inducing the calcium release from endoplasmic reticulum, and is reducing the activity of ATP and acetylcholine channels. And this is significantly affecting the maturation of Schwann cells in vitro as well as in vivo. And M3 activation in myelinating Schwann cells in adult animals is inducing demyelination and axonal degeneration. So that's it. This work was done mostly by Veronica Celli, a senior researcher in our lab, Cristina Angelou, and Jasmine Correale. So the idea of this project that we started a couple of years ago is to use these treats, these receptors that are specifically activated by CNO, to modulate the differentiation of OPCs during development, and also the myelination and the function of these myelinating glial cells in the adult brain. Since, for example, the activation of M3 is promoting OPC proliferation, now we are planning to combine these animals with the Kuprisov model to see if we can manipulate the population of OPCs before and after the myelination, see how this affects the recovery of myelin. And in the peripheral nervous system and also in the central nervous system, this treat can be used to induce demyelination, oligoapoptosis, and axonal degeneration without affecting other cell types. So this is interesting because number one is a model that can be used to modulate the development and the activity of glial cells. And in the other hand, it's a nice model to understand the intracellular pathways and how calcium signalings are regulated by excitatory GPCRs like M3 or inhibitory GPCRs like M4. Okay, thank you very much for your attention.
Thank you very much, Pablo. We are again behind the schedule. So again, we have 2 short questions, 2 short answers.
Can I ask a question?
Yes.
Yes.
Oh, Pancho.
Can I ask a question? Yes. Super. Thank you, Pablo. Very good talk. Very interesting. I have just a short question, very short. You said that if you modulate the activity of this HM4DI, you have alterations in the myelin producing genes and the myelination itself. So the question is, do you have? I mean, is it possible to evaluate whether this molecule is involved somehow on myelin remodeling, and in this way have an impact on motor skill learning, like the functional behavioral output in this case, like shortening the myelin sheath? But the thickness is relatively clear because of your G ratios. But talking about myelin remodeling, yes, that's the question.
Yeah, this is something that we are planning to evaluate in electron microscopy. So far, we know that activation of M4 in mature oligos in the adult brain is increasing the expression of myelin proteins, MVP, PLP. But intracellular signaling, we know that at the same time, the M4 is changing the activity of some calcium channels. So, and by doing so, probably it's also changing how oligos are responding to the electrical activity of axons. The next step will be to check the intracellular pathway. We are planning to do RNA sequencing from OPCs from oligos, for mature oligos expressing the inhibitory drip, the M4. And we have to finish the analysis of the electron microscopy to see if there are changes not only in the G ratio, also only in the length of the internodes, and as you say, in mining remodeling. But yeah, this will be the next step. Yeah.
And the behavioral thing, like motor skill learning, if this.
Oh, yeah, we are planning to do. We have some rot data in the M4, but the animals are completely normal, so apparently motor coordination is fine, right? It's increasing myelin. We didn't find any ectopic myelin in the M4, so there is increasing MVP, PLP in normal areas, but this is something that again we have to check with electron microscopy. The behavior of these animals is normal. They are running fine in the rods. We are planning to do a catwalk analysis. This is a little bit more sensitive to see motor coordination in these animals. But yeah, this is in our list of experiments to do.
Super, thank you.
Thank you.
Thank you very much. Thank you very much, both. So thank you, Francisco. Thank you, Pablo. Wonderful talk, both. So we'll go for the next speaker. Next speaker will be Vanessa Mattera. Vanessa is a PhD, obtained the PhD in our institute, in our lab, and now she is running a postdoc fellow. So she will talk about transferrin containing extracellular vesicles, accelerate remyelination in a demyelinated model. Thank you, Vanessa.
¿Acá? Ah, acá. No. En el PowerPoint. Sí, pero mi PowerPoint está en modo presentación. Acá lo tienen que hacer. Sí. Esto es acá. Ahí, eso. Este es mi. El de arriba. Ahí está.
No dejar de compartir con el PowerPoint.
Porque estamos viendo la tuya. Entonces salís de ahí, lo pongo compartir pantalla.
Ahora sí, dale. No, pero para lo tengo a Rivera, no quiero a Rivera, quiero a...
Si no se me pasa por acá y no se me
No, pero
Para,
para.
La pasamos de la computadora tuya, Nicolás.
Bueno, dale.
Dale. ¿Vos la tenés ahí o no?
Para no, tendría que ser para algún modo para que vos salgas.
Y si la pasamos de acá sin Zoom, ¿qué pasa? La pasamos de acá sin Zoom.
Sí, sin Zoom.
Participantes. Te estoy buscando a vos.
No, porque se desconectó esta computadora, por eso te dije que lo hacemos sin Zoom.
Yo creo que va.
No, poner el PowerPoint acá, ¿lo tenés acá?
Sí.
Hacelo desde acá.
Es Mattera.
No,
Seguro.
está ahí.
Si es acá, presentación GLIA Plus Mattera. Es ahí.
Venga acá.
Ahí está.
Aquí.
Ya está.
Bien.
Necesito conectar el... ¿Me vas a sentar ahí?
Anda pasando con esto, con la flechita.
No, voy a conectar el...
Es que no tenemos que conectar más nada.
Ah, ¿no hay lugar?
No, por eso. Dale con esto, una flechita.
Okay, now we are ready to start. Well, before I start, I'd like to thank the organizers, Juana, Jorge, and Patrick, for inviting me to give this talk. It's a truly honor for me. And sorry, one second. Well, I'd like to start this presentation by telling you a story. The first image that I will show you was published by Pan and Johnson that in 1983 were studying at McGill University the effect of the transferrin receptor during the maturation of reticulocytes. And they observed that during this process, the concentration of the transferrin receptor was decreasing until it was no longer detectable. And that observation led them to ask a fundamental question. Where is the transferrin receptor going? And they found it being released in these membranous vesicles that we know today as extracellular vesicles. And at first, it was postulated that this was a mechanism from the cell to get rid of unnecessary components, like a cellular waste disposal mechanism. But nowadays, we know that this is not the case. And this represents an additional mechanism for intercellular communication. And they will mediate signals not only during physiological but also during pathological conditions. And to illustrate this second concept, I'd like to show you the second image. This is the second image is an electron micrograph of what it seems to be a dark matter cell. And this cell is in the context of a myelin lesion induced by a desolecitin injection. And as you can see in the magnification, this cell seems to be releasing EVs within this pathological process. So in the work that I will show you today, I decided to use extracellular vesicles as a tool. So this will be the talk, a more applied talk than the previous talks that we've been hearing. As I said, that I decided to use extracellular vesicles as a tool to transport transferrin to the CNS using the intranasal route. And I studied the demyelination process using a demyelination model used by glucosome. And when I started my research, I had to decide which vehicle I wanted to use for my research. And EVs immediately got my attention. Not only because they were an emerging tool at a time, but also due to their unique advantages. And the most compelling feature for me is their low immunogenicity as well as their inherent uptake capacity. And that's why I decided to use extracellular vesicles as biological transporters of transferrin. And transferrin is a protein that my laboratory focuses on. This lipoprotein is the main iron transporter in the body and will be synthesized by 4 types of cells. The hepatocytes mainly, the choroid plexus cells, the sperm cells, and also the oligodendrocytes. And the oligodendrocytes has specific iron requirements due to their high metabolic rate. And they will use iron as a cofactor for enzymes that will be involved in their oxidative metabolism, as well as in their lipid metabolism. And as I'm sure you know, those processes are essential to form the myelin sheaths during myelination and also during remyelination. The first evidence, I don't have, the pointer doesn't work here. No, right? Okay, I will do my best. No, I can't. Okay, I will guide you. I'm trying to.
La flechita, la flechita.
No, that is. Yeah. So, okay, sorry, but you will see very clearly. My laboratory, the first evidence of the role of transferrin in developmental myelination when it was intracranially injected in young rats. And as you can see here in this image, the intracranial injection of transferrin increased the number of myelinating axons as well as the myelin thickness. And also, transferrin was able to increase the remyelination after a demyelination injury induced by pyruvate, as you can see here. They tried also less invasive ways to deliver transferrin into the CNS using the intranasal route. And they proved that the inhalation of transferrin increase or repair the weight and matter damage after an hypoxic ischemic event. But the amount of transferrin that they had to use intranasally to reach the brain parenchyma and exert its effect was very high due to the protein degradation phenomenon that is very typical in this route. This is where my work comes into play. My research focuses on investigating ways of deliver transferrin intranasally while protecting it from the proteolytic degradation, as well as ensuring its efficacy and also stability. So I organized this talk based on the questions that I did myself during my research. The first question was, how can I isolate EVs and ensuring functional vesicles suitable for its use as biological transporters? And the other question was, what is an effective strategy to load transferrin into EVs, and if this system is suitable for intranasal administration? So first, I performed the isolation and the characterization of the EVs. I isolated EVs from different sources and using different methodologies, but I will show you the EVs that I isolated from plasma mainly. The technique that I chose to isolate it, the plasma EVs, was size exclusion chromatography. And this technique will allow to efficiently separate the EV fraction from the other contaminants that are present in plasma, and also will allow me to maintain the structural integrity of the EVs through this gentle separation. So I did the size exclusion chromatography. I obtained the different fractions, so now I need to know where are my EVs. So I did a molecular characterization of the different EV fractions, trying to localize the signal of the EVs. In this case, I use C63 analysis. C63 is a very common marker for extracellular vesicles because it's a tetraspan that is expressed in the membrane of the vesicles. And also I checked for ALIX. ALIX is a very commonly expressed protein that resides in the lumen of the extracellular vesicles. As you can see in B, I saw the signal, detected the signal of C63 in the fraction 2, 3, and 4. I put the fractions and I tried to see a specific enrichment of these EV markers compared to the original source. As you can see, there's an enrichment of these EV markers in the EV fractions. And I was also looking for ways to load transferring into EVs. So I searched for the presence of the transferrin receptor in the membrane of the EVs as a way to load transferring through the binding of the transferrin receptor. And I found it, as you can see here. Also, TFR1 is specifically being enriched in the membrane fraction. And as you can see in B, C63 colocalized with the signal for TFR1. And I also checked the structural integrity, the purity, and the size of the EVs by SEM and ELS. I chose to do a SEM to check the topology of the EVs after the isolation method. And I also analyzed the isolated cellular vesicles by dynamic light scattering. And this technique will allow me to see well, the hydrodynamic size of the EVs, and also will allow me to check the structural integrity and the purity by seeing other populations. The technique will detect other populations that will be present in the suspension. And as you can see here, the set average size is in the range for the extracellular vesicles, and also there's no other population present in the suspension. And then I tried passive loading strategies to bind transferrin into the EVs. For that, I used a preloading approach, incubated transferrin with cells with a neuralial cell line or N93, and taking advantage of the intracellular machinery of the cell to obtain already packaged or already transferrin loaded EVs. And I also did a post-loading approach, conjugated the plasma EVs with the transferrin. And these are the results, as you can see, after the treatment with transferrin. The EV fraction, detect the EV fraction in the presence of transferrin, as well as with the post-loading approach. And I also incubated with transferrin hexafed and I measured the fluorescence intensity. So I detected transferrin being loaded into my EVs. I performed a DLS to check the hydrodynamic size after the loading of transferrin into the EVs. And as you can see here, there's an increased size in the EVs that have been loaded with transferrin. And I also measured this parameter, which is the set potential. And the set potential is a measure of the magnitude of the electrostatic forces of attraction and repulsion in a colloidal suspension. And it's a way to determine the stability of the particles. And also can give you, provide you information about the charge of the molecules. So this increase in the cell potential after the binding of transferrin into EVs is telling me that the stability of the suspension is increased. And also, the fact that the EVs has a negative charge is a positive parameter. Mostly due to the fact that I will give an intranasal administration, and in the nasal cavity using a negative charge. So this way, my EVs will not interact with the mucin. Well, then I wonder if I already isolated EVs, I characterized them, and I know them with transferrin. Now I wanted to know if EVTF can be internalized by the oligonucleotide cells, and what are the cellular trafficking pathways. So to that, I isolated the plasmids and I incubated with transferrin texahead and also with a cell tracker, which is a lipophilic type. And I isolated the EVs by sex, as usual. I did the ultracentrifugation to concentrate the EVs, and I obtained EV, TFTR, and EVCT. And the controls are as much important as the experiment itself. So I incubated transferrin texahead and the cell tracker in the absence of EVs to try to see in which fraction I was obtaining the free non-labeled dye. And as you can see, the fluorescence intensity is being obtained, or is being measured after the fraction 6. I put the fraction 2, 3, and 4, and I incubated with the cells, and I saw no internalization phenomena in those fractions. And another very important control is to check the presence of lipoproteins that are very abundant in plasma. So you can have lipoproteins in your EV fraction. So, and also the lipophilic dye can bind the lipoproteins. So if you're studying internalization, this is a very important control. So as you can see, there's no lipoproteins in my plasma EV fraction. And then I was wondering if the internalization was a time-dependent process. So for that, I worked with an oligonucleotide cell line. And I incubated first EVCT at different time points. And I saw the maximum internalization at 2 hours. And here in E, you can see this is the orthogonal view to check that that EVCT is being internalized by the cell. I did the same, but with EVTFTR, and as you can see here, in D is at 2 hours. I saw that you can see many internalization phenomena. In E, you can see that this EVTFTR is effectively being internalized by the oligonucleotide cells. And also, I incubated the cells with TFTR alone. As you can notice, the signal, the fluorescent signal for TFTR is very different from EVTFTR. I think that is due to the magnification of the signal, because many molecules of transferrin are being binded into the EV membrane. So then I was wondering about the internalization pathways. So to demonstrate by which pathways are entering EVs loaded with transferrin, I used VTFTR and I incubated the cells in the presence of different pharmacological inhibitors to block the different internalization pathways. And I blocked the macropanocytosis, clathrin-mediated endocytosis, and also lipid mediated endocytosis. And these are the results. The first thing is my negative control. Of course, I adjust the background signal to this condition. The second one is my positive control is EVTFTR. And as you can see, there's many internalization signal. And these are the results with the different pharmacological inhibitors. And as you can see in the quantification, EBTFTR is entering by every route that I analyzed, but it seems to be entering mainly by clathrin-mediated endocytosis and also lipid raft. And then I was wondering about the intracellular trafficking. So to that, I incubated the cells with EBCT and also with EBTFTR and try to see colocalization events with markers for the early endosome, the recycling endosome, and also for the lysosome. And those are the results. The first 3 lanes are the results of colocalization analysis with EBCT, and with the first marker is the early endosome, then the lysosome and also the recycling endosome, as you can see, they is colocalized with the lysosome. But when I incubated the cells with EBTFTR, I saw the colocalization signal in the early and in the recycling endosome. I couldn't find EBTFTR in the lysosome. So that means that EBTFTR, once is internalized, will suffer different intracellular localizations. The EBs will go to the degradation, will be degraded, and TFTR will be most likely recycled. After the mechanistic studies, now I know that they can enter into the cell. I know a little bit more about EBTF. So now I was wondering about, well, is there any potential in vitro effect on the oligodendroglial maturation? So to that, I started working with primary cultures of oligodendrocytes. I studied the dynamics of the OPC maturation using TGF receptor alpha for the OPCs and MVP for the mature oligodendrocytes. The first MVP was positive control. I use a GLIA-defined medium incomplete without transferring in this case. The second lane is the OPCs with the EB alone treatment to see if the EBs by themselves can exert any maturation effect. The third one is my condition of interest, EBTF. Indeed, you can see that it's transferring, but it's free counterpart. The free counterpart without the EBs, to see if there's any benefit of using EBs, particularly in vitro. And also, the last condition is transferring in a higher amount. This is the same amount of transferring that we use in the laboratory to induce the maturation of the oligodendrocytes. These are the results of the quantification. As you can see, the EBs by themselves cannot exert any effect in the maturation of the oligodendrocytes. But in every condition that I used transferring, there was a decreased number in a decreased number of TGF receptor alpha positive cells, and this was concomitant with an increased number of MVP positive cells. But if you pay attention to these two conditions, the presence of EBTF conditions seems to be maturing more the oligodendrocytes that its free counterpart. So I thought that was interesting that EBTF is capable of mature the oligodendrocytes a little bit more. So I checked this immunohistochemical analysis by Western blot, and as you can see, I saw the same tendency. Finally, this is just to show you that the primary cultures of OPCs are able to internalize EBCT. And then I moved to the in vivo. The next thing that I was wondering if EBTF is able to reach the brain parenchyma when it's administered via the intranasal route. And also I was wondering about the biodistribution and which cell types do they target. So to that, I demarinate animals for 5 weeks using a 50% diet. And I performed the nose to brain delivery of EBT-FTR and also EBCT to study the biodistribution of the bees in the brain parenchyma. So I study different areas, starting by the olfactory route, trying to reach a more posterior part of the brain. This is the more posterior hippocampal zone. As you can see, I analyze the presence of first EBCT in the brain of these animals, in the OPCs in particular. As you can see, at 6 hours post intranasal administration, EB have reached the brain parenchyma, and I also localized the signal of EBCT being internalized by the OPCs in all the brain regions. And I also I did the same with EBT-FTR. And in this case, I analyzed the corpus callosum, which is my region of interest to study the reimagination. I saw that EBT-FTR is capable of reach the brain parenchyma and also reach the corpus callosum, and it was internalized by the OPCs, the microglial cells, and also the astrocytes. And finally, well, I was wondering if EBTF was able to exert any acceleration in the reimagination process, so that use a Hubrisson-induced reimagination model. I was also wondering if there's any advantage in using extracellular vesicles over the same amount of soluble transferrin. Finally, if these extracellular vesicles are protecting the cargo from the proteolytic degradation along the intranasal delivery route. So to that, I use a good model of reimagination by Hubrisson, giving a Hubrisson diet during 5 weeks to induce the reimagination. At week 13, I performed intranasal administration of EBTF. I removed the Hubrisson at this point to return to the normal chow, and I let reimaginate in animals for 2 weeks. So this is just to mention that in every batch that I was studying the reimagination process, I was also checking that the EVs were reaching the corpus callosum of the animals. And these are the results of the analysis of the dynamics of the reimagination using markers. Of course, I use NG2 and PGF receptor alpha to analyze the OPCs, and MVP and Sudan Black, which is a lipid stain, to analyze the mature lymphocytes. The first lane is my control of spontaneous reimagination. As you can see, there's many OPCs that have proliferated and migrated to the site of the lesion, trying to making an effort to repair the reimagination. As you can see, my second condition is the EVs by themselves. There's no change. There's no change in the number of OPCs once you give intranasal administration of the EVs. And also, I didn't see any change when I administered the free counterpart of transferrin non-binding to the EVs. But when I administered EBTF, as you can see, there's a change in the number of OPCs, and this increased number of the OPCs by a decreased NG2 positive cells and also PGF receptor alpha positive cells was concomitant to an increased number of increased percentage of the immunoreactive area of MVP and also an increased number of EVs. And doing the immunohistochemical analysis is not enough to talk about reimagination. So to that, I made a structural analysis of the reimaginated axons. And as you can notice here, the presence of EBTF in the corpus callosum of these animals increased the percentage of myelinated axons, and also increased shear ratios that EVTF is capable of increase the myelin thickness, the myelin sheath thickness. So in summary, I developed a novel method to transfer trampoline using extracellular vesicles, and this main method was suitable for its administration via the intranasal route. EVTF will enter into the cell mainly via endocytosis, and once internalized, the EVs will localize to lysosomes, while transferrin will be recycled. Also, EVTF intranasally administration is capable of reaching the brain parenchyma, and I localized it being internalized by OPCs, microglia cells, and also astrocytes in the corpus callosum of these imaginated animals. And also, EVTF is capable of accelerating the remyelination process, whereas its free counterpart failed to exert this effect in vivo. So these results highlight the advantage of using EVs as drug carriers using the intranasal route in an imaginated model. So I'd like to thank you, and also my advisors, Pasquini and Correale, Federico Fussi, that is the underlab that works with me, and my colleague, Selena. Thank you.
Any question for Vanessa?
Very nice work, Vanessa. Working with the extracellular vesicles is very hard. I have a question about the in vitro experiments about internalization. We perform loading with cyclodextrin and cholesterol and only cyclodextrin in order to to understand not to understand the RAF involvement in the internalization of the vesicles.
Now that's super interesting. No, I didn't do that, but it would be very nice to check that. Yeah.
Pablo, no vas a llegar. Vení acá, no tengas vergüenza. You try to understand the mechanism why these enter the cell, whether you did like a dose-dependent curve, because it's kind of difficult to understand that we use all methods possible, by the cells in order to be so whether you try to, you have your limitations, by your microscope or whatever you can see, but do you do serial dilutions or something to see whether a mechanism predominates? I assume they're going to be calculation dependent.
It's been reported that the EVs can enter by every endocyte in endocytic pathway, lipid, RAF, and also clathrin. No, I didn't know the internalization by doing dilutions.
Okay, the other question is why you have your controls that are EVs from your oligo, OLN 93 that are not loaded with transferrin.
Yeah.
And you said that when you put them in the nose, as OPCs will receive the vesicles.
No.
All the cells surrounding, the OPCs.
Yeah, I detected in all the cells. Yeah, I can show you.
Okay, no, that's fine.
Okay.
Because I couldn't get that part.
Oh, yeah.
So there's no specificity at all.
I didn't make any quantification of that because mainly due to the fact that I was using, if you work with a super resolution microscopy, I think that it's more suitable for make this kind of analysis, a quantification saying, okay, this cell type is internalizing this, and this, and this is the quantification. But in my case, the question that I did to myself with the tools that I found available was, is being internalized or no? So that's like or what? So I analyzed OPCs, microglia cells, and the astrocytes, if they were internalizing EBTFTR, and I saw them being internalized.
Just to finish, the question is, you can use a CHO cell, along with a bunch of transferrin, and put those on the mice, and you will have the same result or not?
No, I didn't try that because I wanted to use plasma EVs from young animals because you don't know the outcomes by using EVs from cell line. So maybe the direction because I understand the point. You use the cells, you have a bulk of EVs, you do that, you give the international administration. But in my case, I decided to use extracellular vesicles from young animals, healthy animals, because I didn't like the idea of using EVs and with a NASA administration to the brain. So I don't know if the direction will change or not. I didn't try it. Just it was a choice idea. Yeah, of course. Yeah, it was a hard work. Yeah, thank you.
One important point is, if you use soluble transferrin, that this was done by Mariano Guardia Claus in 2010, 2012, you have a similar effect, but you need 15 to 20 times more transferrin that you use the loaded vesicles. So you can use soluble molecules, but with the loaded vesicles, you can reduce the amount of molecules that you need to have the same effect. Even in this graphic, Vanessa showed that with 20 nanograms, you have the same effect as with 300 nanograms soluble.
No, I get the point. Just I want to be able to. But then at the end of the day, as shown go to the license, we don't have receptor recycling or things like that. So basically, that's the thing that we just the iron itself.
Yeah, we don't know any other side effects. In fact, that's the problem of working with extracellular vesicles, that there's so much things about the biogenesis and the composition that we don't know. So I understand the point. It would be interesting if for example, the homing and the localization of the suspension, if I use a different type of EVs. I'm sure that they can change if the composition changes. So that's why I stick with the plasma EVs from healthy and young animals. I don't want to do nothing crazy with that, but it would be interesting too. Thank you.
Any other question?
Paula.
I simply wonder whether you have thought how to make this work generate products that can be used in therapy. How you would move forward into human vesicles.
Using transferrin or like a platform using EVs in general? Okay, with a platform, again, if we can think, we want to think about a general platform using extracellular vesicles, we can think about doing chemistry, for example, which implies a chemical modification of the structure of these to load any molecule that you want to bind to them. It's very complicated, but there's some papers that have tried that. Also, with transferrin, the problem is the yield of the bees is very low. And there's some, I don't remember, it's a flow, tangential flow, technique that is improving the yield of TVs. So we are not now obtaining more amount of TVs, but it's complicated. And to apply to make a clinical trial or with extracellular vesicles, I think it's not the time to talk about clinical trials. I think it's time to study the biogenesis of the extracellular vesicles and make an animal that with inhibit secretion of these. Once we develop an animal that you can inhibit, you can prove by the negative the necessity of release, there we can talk about clinical trials. I think so. That's my opinion.
One of the questions of the pharmacy is how you will escalate this for a single individual if you need to prepare a vesicle. So the possibilities to use polyethylene glycol vesicles, that is a polyethylene glycol that is approved by FDA for human use. So now the later coming, how you load this polyethylene glycol vesicles, but it's something that is possible. Or how Vanessa say, increase the number of vesicles that you have by tangential technique. But for the practical point of view, to use synthetic vesicles would be easier for escalation.
Thank you.
All right. Okay. Okay.
Sorry, I have a couple of announcements. For the speakers and chairs, I invited to follow me. Sorry, but I have to tell you, follow me to a place for a sandwich and a beverage. The other people that are not registered have to be registered because we are giving all the people a voucher because we can buy in the place where you are in the school. You can buy whatever you want for lunch. Okay, there is no people registered. Go because we have lunch free today. Okay. Wow. Thank you. Thank you to the International Society for your Chemistry that give us a lot of money for help the students. Okay. So be here sharp at 2:30 exactly because we have to start. And I beg to the chair to ask the speaker